Aging is a universal experience, yet its causes remain one of science's greatest mysteries. We see aging everywhere, in people, animals, and even plants. Senescence, the gradual loss of function, affects nearly all living things. Our bodies become less resilient, organs less efficient, and recovery from illness slows. But why do our bodies, so capable of growth and repair in youth, break down as we age? Aging is not a single process, but a complex web of molecular damage, genetic changes, and cellular failures. To understand aging, we must explore the world of cells, proteins, and evolution. Join us as we unravel why our bodies are built for a finite lifespan, and what that means for the story of life itself. To understand aging, we must look inside our cells. Our bodies rely on trillions of cells, but over time they accumulate damage. DNA is constantly under attack from the environment and metabolism, and repair systems aren't perfect. Errors build up, leading to malfunctioning cells and increased disease risk. Protein quality control also falters, causing toxic clumps that contribute to diseases like Alzheimer's. Communication between cells breaks down, fueling chronic inflammation, inflammaging, and age-related diseases. These failures are deeply interconnected, creating a cascade of decline. Aging is, at its core, a slow loss of order and function at the cellular level. Understanding these processes is key to unlocking the secrets of aging. At the ends of our chromosomes are telomeres, protective caps that prevent genetic damage. Each time a cell divides, telomeres get shorter, like the tips of shoelaces wearing down. When telomeres become too short, cells stop dividing or self-destruct to prevent instability. This limits tissue renewal and repair as we age. Some cells, like stem cells, use telomerase to rebuild telomeres, but most cells do not. This trade-off helps prevent cancer, but also contributes to aging. Telomere shortening is a fundamental reason our bodies lose regenerative power over time. It's a biological clock that protects us in youth but costs us in old age. The balance between renewal and safety shapes how we age. Our bodies are constantly working to repair themselves at a cellular level. However, sometimes cells become too damaged to repair effectively. When cells reach this point of irreparable harm, they enter a state called senescence. This is a critical process in our bodies, acting as a protective mechanism. Senescence involves permanently stopping cell division. The cell essentially shuts down its ability to replicate, preventing it from creating more damaged cells. This halt is a key characteristic of senescent cells. In our younger years, this process is highly beneficial. It protects us from cancer by halting the replication of damaged cells that could potentially become cancerous. Senescence acts as a vital safeguard, ensuring that compromised cells don't proliferate and cause harm. But as we age, the effectiveness of this process shifts. Senescent or zombie cells begin to accumulate in our tissues and organs. This accumulation occurs because our immune system weakens with age, becoming less efficient at clearing out these damaged cells. The body's natural cleanup mechanisms become less effective. These senescent cells release harmful inflammatory signals into their surroundings. These signals are a complex mix of molecules that can disrupt the normal function of nearby cells. This constant barrage of inflammatory signals disrupts nearby healthy cells, leading to cellular dysfunction and ultimately tissue damage. The spread of dysfunction creates a cascade of negative effects. The result of this accumulation and signaling is chronic inflammation and progressive tissue breakdown throughout the body. This is a key driver of many age-related diseases. This process is a significant factor driving diseases like arthritis, where inflammation causes joint pain and stiffness. It also contributes to atherosclerosis, the buildup of plaque in arteries, increasing the risk of heart disease and stroke. Due to their far-reaching effects, senescent cells are now seen as a direct cause of aging itself, not just a symptom of the aging process. This realization has revolutionized how we approach aging research. Scientists are actively developing senolytic drugs, which are designed to selectively clear these senescent cells from the body. These drugs have shown promising results in animal studies, demonstrating the potential to alleviate age-related conditions. The removal of senescent cells has been shown to improve overall health and vitality in preclinical models. Furthermore, studies have shown that targeting senescent cells extends lifespan in mice, suggesting a potential for increasing longevity. Targeting senescence represents a potentially major breakthrough for promoting healthy aging and extending health span in humans. The fight against the detrimental effects of aging may very well hinge on our ability to effectively eliminate these toxic senescent cells from our bodies. Mitochondria are the powerhouses of our cells, converting food into energy. As we age, mitochondria become less efficient and leak damaging free radicals. This oxidative stress damages DNA, proteins, and the mitochondria themselves, creating a vicious cycle. Energy production drops, especially affecting organs like the brain, heart, and muscles. Mitochondrial dysfunction is a central pillar of aging, impairing repair and accelerating decline. Exercise and caloric restriction can boost mitochondrial health and promote the removal of damaged mitochondria. Maintaining our cellular powerhouses is crucial for vitality in old age. Supporting mitochondria may be key to slowing the aging process. Aging, with all its associated ailments and decline, seems inherently detrimental. It brings frailty, disease, and ultimately, death. So a fundamental question arises. If aging is so harmful, and evolution is a process that favors beneficial traits, why didn't evolution equip us with the ability to live forever? Why hasn't natural selection weeded out the genes that contribute to our decline? The answer lies in understanding that evolution's primary focus isn't on individual survival at all costs. Instead, evolution favors traits that maximize reproductive success, ensuring the continuation of a species through future generations. It's about passing on genes, not endless survival. In the wild, most animals don't live long enough to experience the full effects of aging. They die from external threats such as predation, accidents, disease, or starvation long before old age sets in. Consequently, natural selection exerts its strongest influence during the early stages of life, particularly during youth and reproductive years. Several theories attempt to explain why aging persists. The mutation accumulation theory suggests that harmful mutations that manifest late in life, after an organism has already reproduced, effectively escape the scrutiny of evolutionary pressure and gradually build up within the genome over generations. Another compelling explanation is antagonistic pleiotropy, which explains that some genes can be beneficial early in life, promoting growth, development, and reproduction, but these same genes can have detrimental effects later on, contributing to aging and age-related diseases. In essence, nature accepts the trade-off, prioritizing early reproductive success over long-term survival. Furthermore, the disposable soma theory says that organisms have a limited amount of energy to allocate between two essential functions, reproduction and body maintenance, including repair and defense mechanisms. Evolution favors allocating just enough resources to repair the body to ensure successful reproduction. Investing in perfect indefinite maintenance would require an enormous energy expenditure, making it too costly and inefficient from an evolutionary standpoint. Therefore, bodies are essentially considered disposable after their reproductive prime has passed. Ultimately, aging is the result of evolution's primary focus on passing on genes to the next generation, rather than prioritizing individual longevity. Our finite lifespan, with all its inherent limitations, is simply a byproduct of these fundamental evolutionary priorities. The underlying logic of natural selection, therefore, profoundly shapes why we age and why immortality remains an elusive dream. The question of why we age has captivated scientists and philosophers for centuries. One perspective suggests that aging is a programmed process, a predetermined sequence of events unfolding according to a biological timetable. This viewpoint posits that our bodies are designed with a built-in obsolescence, like a machine with a limited lifespan. However, the programmed aging theory is not universally accepted. In fact, most scientists disagree with this notion, presenting compelling evidence that challenges the idea of a preset aging clock. The prevailing view is that aging isn't governed by a genetic death clock ticking away in our cells. Rather, it's the result of imperfect maintenance and the accumulation of damage over time. It's not an intentional design flaw, but a consequence of the wear and tear our bodies endure throughout life. Several influential theories, such as mutation accumulation and disposable soma, support this perspective. These theories elegantly show how aging emerges not from a deliberate plan, but from evolutionary neglect of late-life health. Natural selection favors traits that enhance reproductive success, often at the expense of long-term health and longevity. Essentially, our bodies are primarily built to last through the period of reproduction, ensuring the survival of our genes. Beyond that reproductive window, the selective pressure for maintaining perfect health diminishes, and our bodies are not designed to last forever. There are rare cases in nature, such as the Pacific salmon's dramatic and programmed death after spawning, where organisms are seemingly designed to die after reproduction. However, these instances are exceptions to the rule, representing specialized evolutionary strategies rather than a universal principle of aging. For the vast majority of animals, including humans, we experience unprogrammed aging, where damage to our cells and tissues gradually accumulates over time. This accumulation occurs because evolution essentially stops caring about our well-being after we've successfully reproduced and passed on our genes. The genes that do influence lifespan are primarily those that control repair mechanisms and stress resistance, helping us to cope with the damage that inevitably occurs. These genes are not directly programming aging itself, but rather modulating our ability to withstand its effects. Therefore, the prevailing scientific consensus is that aging is fundamentally the cumulative failure of maintenance processes within our bodies, rather than a predetermined and deliberate program. Ultimately, our longevity depends on how robust and efficient our cellular repair and maintenance systems are at combating the constant barrage of damage. In essence, aging is an unintended consequence of evolution's priorities, a byproduct of the natural selection process that favors reproductive success over indefinite lifespan. Some animals defy aging, offering clues to longevity. The hydra, a tiny freshwater polyp, shows negligible senescence by constantly renewing its cells. The immortal jellyfish can revert to its juvenile form, effectively resetting its age. Greenland sharks live over 400 years, and ocean quahogs can surpass 500. Naked mole rats live 10 times longer than similar-sized rodents and resist cancer. These species have unique adaptations: robust protein stability, powerful DNA repair, and slow metabolism. Studying them reveals that aging isn't inevitable for all life. Their biology may hold secrets to extending human health span. By learning from nature's outliers, we can imagine new possibilities for aging. Longevity is not just about time, but about maintaining function. Despite progress, aging science is full of mysteries. We know the key players: telomeres, mitochondria, senescent cells, but not how they all connect. It's hard to tell cause from effect. Are changes like DNA methylation a cause or a consequence of aging? Lifespan varies wildly across species, and we don't fully understand why. Unlocking these secrets could help extend human health span. Can aging be stopped or reversed? Early experiments show promise, but risks remain. The dream of reversing aging is closer, but still far from reality. Aging is a complex web of genetics, environment, and chance. The quest to understand aging is a quest to add life to our years, not just years to our life.